This episode is supported by two companies invested in advancing GPCR research, GeneTex and Eurofins DiscoverX. GeneTex is building a rigorously validated GPCR antibody portfolio and is inviting you, the community, to challenge it. Request a free sample and put it to the test. And Eurofins DiscoverX delivers validated GPCR assay solutions designed to hold up from early discovery all the way to regulatory review. Links to both are in the show notes. And now, let's dive into this episode.
Hello, everyone. This is Amina from Dr. GPCR, and today I'm very excited to be recording this podcast episode with Dr. Beatriz Blanco. I'm very happy to have her on the podcast. Bea, welcome to the podcast.
Hi, everybody. Thank you so much for inviting me. I'm very excited to be here.
Very excited to have you on. Before we start with your introduction and you telling us who you are and what is it that you do, let's start with an icebreaker. If you weren't a scientist, what would you be doing?
I think if I wouldn't have been a scientist, I would still stay in the same area because I also wanted always to be a doctor. So I guess I would be a doctor, a neurologist or a cardiologist or something like that, doing surgeries.
Still doing scientific hands-on work.
Yeah, hands-on in the patient. So that's what I would do, I guess.
And what is it that you do, Bea? I'm curious to hear a little bit more about what you're doing today, but also about your trajectory that led you to where you are.
So yeah, I come originally from Spain, and I did my studies till the bachelor in Spain. And then I was looking for other possibilities to do research abroad because I was always curious to see if in other countries research was different, was better, because as you might know, also Spain always suffers a little bit with resources for research. So I decided I wanted to move somewhere where this was not that much of a problem. And then I looked for, in Europe, we have what is called Erasmus programs. So you can do like half a year or a year doing your studies abroad. And I decided to move to Germany. Then I did my Erasmus during that year, and I stayed for my master's as well. So I did my master's thesis in neuroscience here in Germany. And then, since I was still curious to see what happens abroad, I moved to the US for my postdoc once I graduated here to New York, to Columbia University. And I continued in neuroscience, but with neurodegenerative diseases. So I changed a little bit. I wanted to explore a bit what was working with disease. And I work with ALS. So I stayed there during my postdoc doing research on that. And after that, I moved back to Germany because my husband is German. So we decided to move back here to Europe. And I ended up here in Leipzig in the Department of Professor Langenhan. You know him because you have also interviewed him here for the podcast as well. Yeah. So yeah, I started here, and now I'm a group leader here. So I have my own group here in Leipzig.
Thank you so much for walking us through. I'd be curious to hear about how was it moving from, Europe, first of all, moving out of Spain to a different country in Europe that speaks a completely different language. They're not even rooted in that and in the same, they don't have the same roots. And then I'd love to hear more about what did it look like at the time for you to move from Europe to the US? The reason I'm asking this is because a lot of people in the audience typically are trainees and postdocs that are potentially thinking about what's next for them. So every perspective is an additional point of view for them.
Yeah, sure. So from Spain to Germany, as you said, the language was the most difficult part because at the end we are all Europe, so it's more or less the same. Also, it's very easy because you don't need visa, you don't need any other paper than your ID, and you can move around Europe. So it's quite easy. It's true that we are very attached to our family, and this was really hard, not only for me, also for my family. So this was really, really hard to tell them at the beginning that I moved for 1 year. My mom was already like, oh, she's gone. And then I said, no, I come back after 1 year. Don't worry, it's only my Erasmus. And then, now it's, I think, 17 years that I'm away from home. So now she's used to it. And from Germany to the US was a little bit more complicated because of the paperwork. First of all, you have to do a lot of stuff to get the visa and contracts and stuff like this that you need to work there as a European. Also, the culture, I found it a bit different than what we have here, but I loved it. So I loved to be there and actually didn't want to move back so early for me. My husband put a little bit of pressure on that. But it's true that, for example, the lab workflow and everything was a little bit different. So how this is a structure there compared to here to Europe. But to me, I really like it and I enjoy it there. So I cannot say anything bad about the US. But it's true that culturally we are a little bit different also.
Yeah, it's an adjustment. And you mentioned, well, moving to New York is already, I think, a lot because it's New York City, and not only the fact that it's US, but it's New York City. I lived in Manhattan as well during my postdoc, and I was at Rockefeller, so on the east, lower than Columbia, and it was moving from Canada to New York was already a shock, because everything moved so fast.
Yes, it's very fast.
And it's noisy and it's loud, but it's also energizing.
One quick thing before we continue. The Dr. GPCR University is built for scientists who want training that serves real discovery problems, with live and on-demand courses within a nonprofit ecosystem dedicated to advancing the GPCR field. All the links are in the show notes. And now, let's get back to the episode.
Yeah, true. I think the US is one thing and New York is another thing, because this city is completely different. So when you move around the US, then you see what real US is, because New York is, as you said, very fast. Everything is very, very fast. But for me, it was fascinating because it was something completely different what I was used to experience here in Europe, even though I come from Barcelona, which is a big city, but it was not the same. So you cannot compare it to New York. But as I said, I really enjoyed it there as well. But you have to adapt. You need to adapt to this rhythm that they have there.
Yeah, for sure. And I think it's a nice experience, especially when you go back to Europe or you go back to your country and you're like, wow, what a big difference there is. Not that it's good or bad, it's just different.
Right, it's different. One of the reasons why we moved here was, for example, because of the kids. When you want to have kids, it's easier here in Europe than in the US. So that's why we also decided it would be a point now to move back to Europe. But it's a different experience.
Absolutely. When you were a child, I'd be curious, because you mentioned if you were not a scientist, you'd be a doctor. But when you were a child, did you always know that you were interested in science, or was that a moment or potentially teacher or family member that kind of influenced you into getting into science and research?
No, always. Since I was very, very small, I always said, I'm going to be in the lab, I'm going to be a scientist. Even though people say, why do you study this? Because in Spain, the situation, they say, why do you study this? You never will find a job. You never will work as a scientist. Yeah. And I thought, well, let me try and then let's see what happens. Thanks God I did it, because since then, thankfully, till now, I'm a scientist. So I'm very proud that I continue and I didn't give up on these studies. But since the very beginning. So I always thought, because if you study medicine, you can also go to research after that. So that's why it was always like, I don't know, shall I study directly biology, because I studied biomedicine at that time, or shall I go to study medicine and then go to research? But at the end, I decided for biomedicine. So that's why.
Here we are today.
Here we are.
And where do GPCRs enter into the picture for you during your training?
So they enter quite late, because when I started with my master thesis, I was working in a very simple question in neuroscience, so to study synaptic proteins. This was what we did, and I continued with this during my PhD. And that's why during my postdoc, I said I want to go to disease to see a little bit something different from the neuroscience that I didn't get to see during my PhD. And then when you see like basic science, and then you get this translational medicine that you can apply this to patients and you can work with them, something is missing in between. So there is something that connects both of them. And at that time, I was not aware of it. But thanks to the possibility to start here working in an adhesion GPCR group, I could get my hands on in adhesion GPCRs. And then, since now, I think will be 9 years this year, I'm working on receptors, adhesion GPCRs that are expressed in the fly, so in Drosophila melanogaster. And since I moved back to Germany, then I started with this, and I find it fascinating because you can connect them both things I did previously. So that's really cool.
That is fantastic. And, we're recording this episode now. It will air in a couple of weeks. But our next episode that's coming out in a couple of, in actually 2 weeks, is about studying GPCRs in mosquitoes. So that's also, I think these model organisms are so fascinating because when you think about it, these have been used for a very long time, and people might think, oh, we know everything that we can know out of these model systems, and we don't necessarily need to use them anymore that much. And then come you and our previous guest, and the conversation and the discoveries in these model organisms are still so fascinating.
Yes, I mean, everything you can find in an insect, let's say, because mosquito or Drosophila, even honeybee or something like that, you can always translate it to vertebrates. So that's the beauty of this. Also, it's a simpler system, let's say, but still very powerful. So I did Drosophila during my PhD, and then I changed to mice when I went to the US. And I have to say, I don't miss it because it was taking me so long to get some results compared to the flies. So that's why I was happy to go back to the flies because, you can reproduce them very easily. So it goes really fast.
Yeah, you don't have to wait 6 to 8 weeks and then 12 weeks, depending on what you're treating your mice with, and hoping that they mate, especially if you're making some knock-ins or knockouts and then genotyping them. That's a whole other ball game there. I'd be curious to hear more about what are some of the questions that you're trying to answer using the Drosophila as a model system. And obviously, I will be asking you which one is your favorite adhesion GPCR.
Yeah, the question. So when we work with adhesion GPCRs in vertebrates, when I started here, characterize were only 2 receptors or on the way of characterization, but then they were known to have, or Drosophila is known to have 5 adhesion GPCRs, at least as far as we know. And the other 3 were completely unknown. So we didn't know the function, we didn't know where they were expressed, nothing. And here in the lab, we generated knockouts to start with, or we could manipulate the receptor, and then we could characterize them. And that's what I'm doing at the moment. So I'm working with 2 different adhesion GPCRs. So as I said, I'm a neuroscientist. So one is neuronal. So I work still with neuroscience, but the other one is not, is expressed in the gut. So, and this is also very nice to explore a new organ that I didn't work with it before. It's very complex also. And we try to characterize these 2 adhesion GPCRs to see which function they have, what signaling they do, what can activate these receptors. So we work a lot in vivo with the fly, but we also have in vitro systems that we can manipulate and to try to get these answers.
And I'm assuming these 2 out of 5 adhesion GPCRs are also conserved in humans as well.
One, yes, one is conserved. The other one we think is like primordial. So it's very, very small compared to the other ones. Like when we picture an adhesion GPCR, they have this long extracellular domain. That's why they are called adhesion mostly. But this one doesn't have such a long extracellular part, but still like what is called the gain domain or all the characteristics that an adhesion GPCR might have. But when we try to check phylogenetically where this receptor will fall, we cannot find any family that directly connects it. So we think it's a primordial one, and from there, the other ones might have evolved.
Interesting. And can you share which one is your favorite?
Yes. Well, in Drosophila, we have very funny names for our receptors. I don't know if you heard about them already, but so the 3 new ones discovered, we name them as sauces. So one is Ketchup, the other one is Mayo, and the other one is called Remulate.
Okay.
My favorite one is Remulate because thanks to Remulate, I could get funding to have my a PhD student working on this project. So, and it's also the neuronal one that I just mentioned. So that's why I started from scratch with a previous PhD student that we had in the lab working on it. Yeah. And now I can continue further research on that. And this is amazing because it's all from the very beginning. So that's really, really nice. It's a very difficult receptor, I have to say. It's not an easy one, but which one is easy, right? So
Exactly. And I think with the adhesion GPCRs, we're at the beginning of discover or building the toolkit. Few years ago there were no structures. I think 1 or 2 years ago there were structures coming out, and also the tools. It's not your regular flavored GS or GQ coupled receptor where you have your little assay and then you can run it and screen for molecules. And also the fact that there aren't small molecule tools as widely available as for class A GPCRs, for example, makes it all the more harder.
Yeah. It's hard to find something in these receptors. Takes really long, I would say. But then when you see it, then you forget how long it took for you to go through all these experiments to get there. But thanks to also this structural biology and everything that is coming new, all the tools that we can get nowadays is getting a bit easier. So, or at least we can speed up a little bit the process.
Not having, I never worked with Drosophila. I'd be curious to hear about some of the intricacies of working with such a model system. For example, how long does it take to have a new colony, if you call it that? And what are some of the key methodologies that you use in the lab? I'd be curious to hear a little bit more about it.
So with Drosophila, it's very easy to manipulate compared to mice, for example. So we can generate a lot of lines to study these receptors, for example. And it's relatively fast because after 1 week or a little bit more than a week, you get your new generation. So it goes quite fast compared to mice, like we were discussing before. We apply a lot of CRISPR genetic tools because we want to modify these receptors to see what happens when you don't have them, what happens in the fly, which phenotypes you can see. Then we modify them with tags because, as you know, for GPCRs in general, the antibody collection is not so great. So we decided just to add tags that then we can see where this receptor is located in the animal. Or if, for example, we want to modify and remove some parts of the receptor, we can also do that. Mm-hmm. So we work a lot with genetic modifications of the flies. And then we can directly generate flies in like 2-3 months, new flies. So it's actually quite fast. And it doesn't take so much time if you compare with other animal models.
Yeah, and the readout, is it behavioral, or
so for our projects we have behavior, but this is not the first thing we checked because we are not a behavioral lab to start with. But of course, you always depends where the project is guiding you. You can end up doing behavior as well. So the first thing we did was see where is this receptor expressed, and can we see a phenotype where this is when we take it out or when we knock this receptor out, can we see an effect in any of these pathways that could be there? And at the end, it guides us to behavior, of course, and then we can see if they change. We can study this in larvae, so we can do it when the animal is really, really small and just developing, or in adults. So this you can decide depending on where the project goes or which kind of cells you are interested to look at. It makes it easier to work with larvae or with adults. So this you can decide during the project and whenever you see a phenotype, for example. But we have used behavior, for example, for Remulate because it's expressed in the peripheral nervous system. And for that, in Drosophila, you have a lot of behavioral tests that you can apply and check if you see some differences.
If I remember correctly, Remulate has a human ortholog. What are some of the diseases that this human version of the protein is involved in? I'd be curious to hear a little bit more about more of a clinical, potentially, or disease-related phenotype.
Yeah, so the homolog for Remulate is A2 receptor or A3 as well. And because in vertebrates you have more families than, or in the same group you get more families than in Drosophila, that we have one for all. And so it has been shown that in mice they have vascular malformations or blood-brain barrier problems. And we were also interested to see if our receptor would be, for example, involved in, I don't know, or we would find the receptor in the glia, for example, because since it's blood-brain barrier, Drosophila doesn't really have blood-brain barrier, but something similar to this. And we could see that it's there. And also being able to study this receptor in the fly, we could see that, for example, the peripheral nervous system that has not been checked in this mice, as far as I know, we could see there that they show, so we call it nocifensive behavior. So when they have a nocifensive response, the larvae, when they feel like they are getting attacked. And when we knock out our receptor, for example, this nocifensive behavior, it's increased. So they respond to this kind of, I wouldn't call it pain, but kind of this with when the receptor is absent, for example. So it could be clinically, or going to the clinical part, it would be some neurodegeneration or neuronal pathways that could be studied there if you want to compare it with the vertebrate version.
That is fantastic. And when you mentioned that this receptor is part of this family, I love the naming, by the way. We have such a sense of imagination as scientists when it comes to naming new proteins. I'd be curious to hear a little bit about do we know what it does in the fly, or what is the structure of it? And is this a receptor that was recently discovered? Or what are mainly the steps that led to its discovery?
If we check only the fly, yes, it's relatively recent, not like last year, but we are, for example, preparing our first publication on this receptor now, so it's relatively new. And so it was discovered based on rescreening the genome and checking for characteristics from the adhesion GPCRs, for example, if they have a gain domain, if they have a 7 transmembrane domain, all the characteristics that an adhesion GPCR could have, then they were checked, and then these 3 new receptors were discovered. And from then, we just decided, okay, let's check if we alter this receptor in like a knockout or in any other way, if we can see something and we can study the phenotype that it has, or if we tag it, if we can find where it is. And that's what we are doing right now. So it's not the very, very beginning, because we have worked a little bit on this. Mm-hmm. We try to place also the receptors, because a lot of problems we face normally when studying GPCRs is where exactly is this receptor, because you can say, I think it's in this organ, or I think it's doing something there, but where exactly do you have that? In our case, for example, do we have it presynaptically, postsynaptically, since it's a neuronal receptor? Mm-hmm. In both, is this receptor doing something? This is something we are trying to figure out now and find. And yeah, we are actually at the beginning, I would say, of the discoveries for this receptor.
That's so nice, because when you think about it, usually people say, oh, when you're a scientist, you're the first person that discovers something, and that can be true for a signaling pathway. But when it comes to discovering what a receptor does and where is it expressed in any organism that you're working on, I think it's just such a fascinating and an interesting journey, because you might have some assumptions, but this is the first time that we're looking at this receptor.
Right. It's like there's nothing known at all about this receptor, and then you come into play, or our group comes into play, and then they, piece by piece, they try to, it's like a puzzle, right? So you have to just put the pieces together, and then at the end you can discover what receptor that was there in the fly, nobody ever checked what it's doing, at the end it's doing, and if this could be translated, obviously, because the idea of research always goes to the clinic at the end. Either we like it or not. Basic scientists might say, I'm interested in the question. I'm also interested mostly in the question. But at the end, of course, if it can be applied somewhere, it's also really, really good.
No, I absolutely agree. Considering that this is a relatively new receptor, and at this moment you're working on better understanding what it does in the fly, if you had a magic wand, and let's go into imagination land, and you could develop a tool that would allow you to quickly or better characterize this receptor, what would that look like? And we can be as imaginative as possible.
I actually don't know, because actually antibodies would help a lot. But yeah, just to have a machine, and then, I don't know, put your for example, let's say you have a protein lysate of your receptor and you want to see which ligands this receptor might have. And then you put it there and everything works and everything, it tells you everything. So how this receptor changes when the ligand is binding. So this would be great. But of course, it needs a little bit more work than what we imagine.
Exactly. The reason I was asking this question is because I remembered my conversation with Anthony Boucard, and he said, when I asked him this question, he said, "Oh, I wish I could, go down or size myself down to the size of a GPCR and be able to observe the adhesion GPCR signal." And I thought
that would be great.
There was a cartoon called The Magic School Bus back in the time when they were on a bus with the teacher, and they would go into the body and they would explain all sorts of mechanisms about how the blood flows and the lymph and things like that. And that's the image that came into mind.
Yeah. We also had this like the human body, and then you could walk there inside. So something like that. But we have to be very small for that, I guess. But that would make it very easy then. That will make our job really, really easy. We could just observe and then go back and say, okay, I know now.
Exactly. I know what happened.
Yeah.
During this journey of, research that you've been on for so many years, were there any moments where you said, oh, these are aha moments or maybe got you moments that shaped your trajectory?
Yeah, because when I moved from Spain to Germany, for example, in Spain I did my bachelor's, so it's very general. You don't really get to deep your knowledge in anything. You do the practical courses, well, like every student, but nothing special. And then I moved to a neuroscience lab, and then I said, okay, I love that. I love what I'm doing there. I love protein biochemistry as well. We are here in the biochemistry department, so I really enjoy working with protein biochemistry and doing my master's and PhD later. I got to work a lot on that, so I really like it. That's why I said, okay, I think I want to continue with science. I don't want, because a lot of people after the PhD, even though it was really hard, they decide it's not for them, or they want to change, or they want to go, I don't know, to industry or pharma. And for me, it was more like, okay, now I want to go to the US and I want to move and do something with always neuroscience because it was fascinating for me, but something new. And then in the US, of course, we could apply what we were doing in mice to the first patient with a compound, and this was also great. So for me, to see that our work works or it helps in some way, this was also amazing. And then back here, also when I started with the adhesion GPCR, which was completely a new field for me, and then it was kind of not starting over, but seeing the stuff from a different perspective, because you come from disease and then here you don't really work on disease anymore. You work on a receptor and you want to study what happens there. But then to see all the possibilities that this offers as well was also great. So that's why, because I have to be honest, when I moved back from the US, I was not sure I want to continue in science. I don't know. It was not like clear that I want to stay in the lab and continue with that. And thanks to Toby, of course, that gave me here a position and I could stay here. I'm still here, so it means that I enjoy it.
Absolutely. I think that's very important to stop and think about what is it that you want to do, and then decide where to go from there. You mentioned coming back from New York to Europe, and also you mentioned thinking about what next, lab, bench, or potentially industry. Has it ever crossed your mind to go into industry?
Yeah, a lot of times. Because at the end, working in the lab is also not only what you do here in the lab, you work also at home. And having kids, for example, you have to really put a big effort on doing that because you need to take time wherever you don't have the time, but you need to take the time. And yeah, there were also some points during my career that made me think, do I want to be here? Because not every scientist is treated nicely, I would say, or in a nice way sometimes. Or for example, publication problems, authorship problems, stuff like that. And So yeah, I thought a lot of times, okay, I give up. I don't want to be here. It's too much. I move. I will look for something else. But then, of course, I love to work in the lab. Or now, unfortunately, I cannot be in the lab so often, but because I also have to teach here at the university. But I love to be in the lab. And then it's stronger than anything that you can, or your brain at some point thinks. So I always thought like, okay, I apply for a job, but then if I get it, do I really want to leave? And the answer was always no. So then you have your answer. So
yeah, no, I think that's wonderful. And I'd be curious if you can go back a little bit in time, since we're talking about a little bit about, how you made your decisions, is when you finished your PhD, how did you go about doing a postdoc? Did you apply to different labs? And how did you pick the particular lab that you were in?
Yeah, I applied to, actually, I wanted to work with mice. That was one of my main, let's say, things that I wanted to do, because I did Drosophila, and then I wanted to explore. When I changed completely. Some people always staying the same and continues with the research. For me, it's like, no, I want to change and I want to learn something completely new. I had some hands-on with mice doing my PhD, because the lab next to us, they were all working with mice, and I could do some experiments. So I wanted to really explore that. And then I applied to different labs in the US. I remember I think one was in the NYU, another one was in New Jersey, and then at Columbia. But yeah, I met there with the PI, and I really liked the project. So then I decided for this lab. But yeah, I was looking in different labs, but always disease, of course.
Yeah, that's right. I understand correctly. The topic was pretty much set. You just needed to explore what lab has works on those topics. And also, I think there's also this question of fit. Do I want to be here, or I want to run away? Because those situations also happen.
Yeah, I had, for example, in the lab in New Jersey, I interviewed. I loved the group. They were very nice, but I didn't feel safe where I was. And then I was thinking, like, do I want to come every day here to work when you see security everywhere, because the area was not safe, they told us. And then, of course, you also have to think about not only science, because science is the most important part, but also how you are going to feel going every day to work. And for example, in this case, was safety reasons that I didn't decide for this lab.
Wow. I don't think I've ever heard anything like that. That's scary when you think about it, that you don't want to go there because you mentioned that they told you.
Yeah, they told me. They said, no, we pay you a taxi. You come with a taxi. Don't come walking. Don't take the public transportation. I was like, okay, where did I go?
Where is this lab? Wow.
Yeah, I totally forgot about it. But now that we were going back to when I applied for, I said, no, I don't want to have this feeling every day going to work, and then something and maybe never happens, or could happen anywhere. But yeah.
Yeah. But when they warn you, I think it also adds an extra.
Right. They're like, okay, then let me think about it.
Exactly. Any advice for young scientists entering the field, working on adhesion GPCRs or working on GPCRs in systems like the Drosophila, for example?
I mean, working on GPCRs, it might be frustrating. We had this here with our students also, that sometimes the stuff doesn't work and then you want to give up, because, especially when you do your PhD, I think they are in such a pressure that, they just think, okay, that's it. I don't want to do it anymore. It doesn't work, the experiment. I don't get any results. It's horrible. But don't give up. Everything works at the end, in more or less degree, but at the end, the stuff works. And if they really like science, that's what I would say. Never give up, because it's your passion, it's your job. So they will for sure enjoy it at some point, and they would really, like what they do. Because the most important, I also always talk with my husband, he's also a scientist, and that we love what we do. And we have a lot of friends, they don't like what they are doing as a job. And this is very important, I think.
No, I agree. I think it's very important. And to your point, there's a lot of pressure, especially when you're in a PhD or a master's where you have certain prerequisites that you need to satisfy before you can graduate. But also as a postdoc, when things are not working, it's your career that's in the balance.
Yeah.
You're not evaluated on your effort, you're evaluated on the outcome, which is the papers and the results and that. And unfortunately, there is no journal of negative results, because I think that would be very useful. But at least you could put it there to say, well, we've tried this and this does not work. And I think it would save a lot of people a lot of time as well.
Yeah, exactly. If they would have, you would give the same credit to something that is negative, but doesn't mean that you don't learn anything from it, because yes. Okay, you try this approach and it's not working, fine. Then let's try another one. But you learn that you cannot go through this path, so you can change. That would be really good. I think there are some journals that accept negative data nowadays, which I find really good.
I didn't know that.
But yeah, I think it's called eNeuro or something like that, and they accept negative data, which is fine, or I find it really good, because then, as you said, for the PhD, you have to prove that you did something. And sometimes it's not enough with writing a thesis. You have to have a proof to show, okay, I have this, I did it during my PhD and it's published. And this is very important, I think. Because, with the students, sometimes you also have to change projects, because you are in not only pressure of project, pressure of time. They don't have all the time in the world. I think in the US, it's a little bit better in this way. They can stay longer. At least I've seen PhDs staying longer than 3 years, for example. But here, the funding we get is not longer than 3-4 years. So,
yeah, I think it's also, to your point, it depends on where you're doing your PhD, where you're studying, because I think on the other hand, I think in the US you can stay up to 6 years. Which can be very long, and sometimes you want to leave, but you can't, because there's always things to finish and always experiments to run. Where I did my PhD at the University of Montreal, at the time, they had a prerequisite of 2 published papers. And you needed at least 2 manuscripts. I don't know if they had to be published or submitted, but it was supposed to be 2 full manuscripts. And so there was that pressure of, I need those 2 papers to graduate.
Yeah, and here also you have, depending on the university, they can ask you for more or less.
Yeah.
Where I graduated, I think it was at that time 1 first author already published.
Okay.
So also, you need to have that. Otherwise, you cannot finish.
Yeah,
so.
Yeah, and I think in Europe, it's nice that it is time bound. But at the same time, the 3 years might not be enough because of you might be unlucky with your project. All sorts of things can happen. So it's definitely stressful on that.
Well, the example you have is, the example you have is with Corona, right? So when Corona hit, you lose a year or more. And then we were lucky that they gave us an extension to some of the projects, but of course, it's not the same.
Exactly, and you want to move on with your life and not not get stuck there
at some point.
Yes, absolutely. You mentioned that you teach. I'd be curious to hear about your teaching experience. Do you like it, and do you think all the training that you've had during all these years working in the lab prepared you to teach? Actually,
I mean, it's very different, because I teach medical students at the university. We are in the medical campus, so we teach medical students, and we teach biochemistry. One thing is, I'm not German. I have to teach in German, and this was very challenging for me to prepare. But I like to teach. It's not that the problem is you lose time when you teach. You lose time for your research also, because you have to combine both of them. But to see new students every year is very refreshing, because they are very young, and you see that they are at the very beginning of their career. Some of them also decide to do the medical thesis with us, and then I can teach them in the lab, which I like even more. When they come here, they take a year off, and then they come, and then they do their thesis. And this I really like, when they are here in the lab. And for the regular teaching, I do now seminars for medical students, and it's also I started this year with the seminars. I did practical courses before, and I have fun. So they're nice. I can't complain about my students.
No, that's great. Yeah, medical students usually are very focused, very driven, and it's move through these courses, especially that these are I'm assuming mandatory courses, but it's not going to change their trajectory. So they just want to get the knowledge, learn it, pass the test, and move on usually.
Right, especially for biochemistry, they want just to pass it because it's a preclinic course, and it's not most of them, what they want to do. So,
yeah.
And they are really, really stressed. So they have to do not only biochemistry, physiology, and other subjects, and then it's way too much during the year. So they just want to pass exams and then forget about it and move on. So that's how I see it sometimes.
That's how I experienced it in the past. And anyone I talk to, they're like, no, they need to do their thing and then move on, because that's not going to change much other than them having to pass the course.
Yeah, exactly. Well, and then you find the ones that want to come to the lab and they are curious. And this is fun because then you can show them what you really do in the lab.
Yeah. That's actually the fun part of having students in the lab. Absolutely.
Right.
Are there any conferences that you're planning on going to? Anything exciting you want to share with the audience?
Yes, in September we have the adhesion GPCR conference in Düsseldorf this year, so I'll be there. And here in Leipzig, before there's the GPCR meeting as well that I highly recommend if somebody's interested. Everything will happen the same week, so it's crazy because we are going to be the whole week listening to talks and seeing people we know from, for example, for us, the US or abroad, and they will all come first to Leipzig, then to Düsseldorf. There will be a shuttle bus that will take us to Düsseldorf from here, so it's actually quite nice. And I recommend if somebody is thinking of attending any conference that either adhesion GPCR, which is a bit more specific, so you have to be in the field, but the GPCR conference is also, I think, pretty nice to attend.
I saw, and I love the fact that there is a shuttle between the two, so you don't have to think about it. Because initially when I heard about the two conferences and I actually Google mapped the distance between Leipzig and Düsseldorf, I was like, okay, that's quite a ride there. But having a shuttle bus, it's so nice because everybody's going to stay together, and then you get to talk to people the whole week, basically.
Right. I find it a great idea because then it's like, well, when we were in Mexico, like almost 2 years ago now, was also really fun when we took the shuttles in the morning to go to the conference, and it was really, really nice. So because you are in permanent contact with people there. So
exactly. Yeah. Antonia and I had a lot of fun organizing that. And we told each other we absolutely need that shuttle bus, considering that there was this distance to go from, where it was basically safe to stay and Sinvestav. So we needed to have those shuttle buses. And it was I told him we have to do this, and Anthony was like, yes, we will do that. And then I'm happy that people enjoyed having the bus. Unfortunately, I couldn't make it to that conference. Hopefully, I'll be able to make it to this one in Düsseldorf.
Yeah. For the Mexico, you did a great job. So it was great. We had a super good time there. And you should make it to this one this time because we couldn't see each other in Mexico. So at least here.
Hopefully, we'll make it. All right. Last question. Where can people find you? Do you usually use LinkedIn, or if you have, a new job posting or anything that you post about your work in your lab, where can people find you?
Yeah, on LinkedIn, they can find me with the same name. So it's all my name there, and you can find me. And if you Google also my name, it appears our university webpage, and there you can read more about what we do in the lab, and when we have some positions, something like that also will be announced there. Or in this time, for example, when I had a PhD position, it was in LinkedIn, so people can also see there.
Wonderful. Bea, thank you so much. We will also link these different sites where people can find you on the podcast page. Really enjoyed the conversation, and I hope to see you in person in September.
Yeah, it was really nice. I enjoyed it as well. And I hope to see you there.
All right, see you there.
Bye-bye.